So with that, I wanted to welcome you to the opening basic science session of the AIRRC Community 6 meeting. It's really my pleasure to introduce our keynote speaker this morning, Gunilla Karlsson Hedestam, who is a professor at the Karolinska Institute. And she has done really amazing work on antiviral immunity and immunogenetics, has been a longtime member, active participant in the AIRRC community. And I think almost all of us also know her really nice tool, IgDiscover. And with that, I will hand things over.
Thank you, Steve. And I'd like to thank the organizers. It's really nice of you to have me. I will talk about a couple of different areas that we've been working in over the years. And most of this is unpublished, and it covers both BCR and TCR variation. We've taken a great interest in understanding germline genes and allelic variation. To us, this is sort of where everything starts. It starts with the alleles that a given person has in what sort of response that person mounts, whether it's in the context of infection, vaccination, cancer, or autoimmunity. So I will talk about several aspects related to that. Starting with BCR VDJ allelic variation in a population study that we've done. I will then talk about a specific project where we look at the functional consequences of this, because we often describe this variation, but we don't have so many examples of showing how it really impacts an antigen-specific response. And then I will talk about something that we've done recently, which we find quite interesting, which relates to inheritance patterns, specifically in the IGH locus. And then I will finish with talking about TCR, where we've done a sort of major study recently. And I think everyone here knows that these loci are extremely difficult to capture by the traditional sequencing methods. There is a lot of repetitive elements. There is pseudogenes. You really do not get any coverage with short read traditional sequencing approaches. And you cannot sort of identify alleles based on SNP data alone. So at the moment, there is several efforts in this community using either long read sequencing or germline gene inference, and ideally a combination of these two, to characterize variation in BCR and TCR loci. And I think this is going to be very important for understanding how we all differ. This was highlighted in a commentary we wrote, several of us here, where we said that studies in immune repertoires and germline gene variation really need to encompass multiple populations. It's been quite restricted towards studying people of European sort of origin, and this really needs to be sort of diversified and extended to other populations. So we have done a study where we looked at 80, in fact, 100 individuals, but I'm showing 80 here today, in terms of the BCR germline gene allelic variation. And for TCRs, we have a slightly smaller group, but we have populations that cover different parts of the world. And the goal with this was to, of course, identify novel alleles, but also to determine the overall frequencies of different alleles, and to see if any of these were restricted to some population groups. And also in doing this, we, of course, pay attention to copy number variation and this sort of thing. So this was really kind of something we set out to do many years ago. And I'll start talking about the BCR variation. And this is a project that's been led by Martin Corcoran. I think he knows, most of you know him here. He's been greatly helped by 2 PhD students and 1 postdoc, Sanjana Narang, Matheus Kaduk, and Mark Chernyshev, who's here. And what we did here was that we had donors from these different parts of the world. We applied IgDiscover, so we made IgM libraries, 2 libraries per person, using different primer sets. And we basically inferred the V's and T's and J's in all of these individuals. And we used multiple approaches to validate our sort of allele identification. And I'm not going to go into all of that now because we presented it before, but I just want to kind of show you a summary of what we find. And this is sort of the summary of the number of alleles you find if you look at in the 80 individuals we looked at. And what this tree shows, you cannot read it, of course, but you can see that there is multiple alleles in this tree that have the longer names. And for those of you who use IgDiscover, you will know that this is the name you get with this S 4-digit suffix. And so you can see that actually almost exactly half of the alleles are novel. And that means they have not been previously reported. We're comparing to the alleles that are in the latest release of IMDT. And these are present in all the V gene families. We see 307 alleles in total in this population, of which 149 are not previously described. These are really distributed on all the V genes. So this plot shows the different V genes and the number of alleles we find per gene. And the blue are the ones that are present in IMDT, and the red are the ones that we found in this population group. So I think with the exception of one gene, we find novel alleles. And for some genes, we find quite a few novel alleles. As is known, the genes that have the highest allelic variation, IgD330 and 169. 169 is extreme. I'm going to talk about that a bit later. It's also highly used in the repertoire. So it's relevant, and people often pull out monoclonal antibodies that use this germline gene. In this work, we've done extensive validation, as I said, and that means that we've looked at alleles that are present in more than one individual. We do a lot of targeted PCR and genomic sequencing, Sanger sequencing to validate that the allele is truly present in the genome of the person we found it in. And we also do haplotype analysis, which we find to be extremely useful. We found that most of these alleles have coding changes. Not all, but the majority actually have coding changes. And many of these alleles are population restricted. They're only found in one population, but not in all populations. If you look at how many alleles a person has, you can see that it ranges anything between about 40 to 80 approximately. And this plot is just showing the donor cases on the x-axis and the number of alleles on the y-axis. And if we have 50, 52, depending on how you count, V genes in humans, you can see that the people to the left of this graph are the ones that have high level of heterozygosity, and the individuals to the right are much more homozygote, or they have multiple deletions. So their total number of alleles is much lower. So this is sort of the typical range you see in humans, between 40 and 80 alleles. If we split this into the population groups that we studied, you will see this pattern. And we find this quite interesting and also in a way expected, because the allelic variation is by far the greatest in sub-Saharan Africa. And you can see this sort of reflected in this graph in that generally the sub-Saharan African donors here tend to be more heterozygote and have sort of a high allelic content. So this is in a way what you would expect, but it's interesting to see. And we don't see this so clearly when we look at light chains or TCR alleles, but in the IGH, this really kind of stands out. And this is just to kind of give you the impression of some of the alleles that we found in a population restricted manner. This is not all of them. I just took a sum to fit on the slide, but it gives you an impression of that there's a large number of alleles that are only found in sub-Saharan Africa. And curiously, there are alleles that are only found outside of Africa. And if you look at the Americas, you will see, I will come to that, but in this particular donor groups, we did not have those, we only had Africans, Europeans, Southeast Asians, and East Asians. So this is kind of the picture we're seeing. And what does it mean? So basically, each individual we look in, we see a unique content of alleles. This is just showing one of the V gene families, but it really kind of highlights how personal your allelic content is. And for a lot of the biological projects we're running in the lab, we have this sort of, we consider it important to always do personalized genotyping of the person we're studying. Whether you're studying the development of a lymphoma or a vaccine-induced response or response to SARS CoV-2 or whatever you're studying, we want to start with knowing the germline allele content of that person. And the question then that often comes up is how critical is this? Because B cells typically interact with their antigen mostly via CDR3, which is not all germline encoded. So it's a reasonable question to ask. How important is this? And we had an opportunity to look at this in at least one study that is the one I'll describe here today. In the context of the pandemic, I think a lot of immunologists were now in a position to get interesting samples and ask basic questions, because you had a virus that's basically sweeping through the population, and you could start asking questions about populations and variations, genetic variations, and the impact on the response. So the project I'm going to describe in relation to functional consequences of this allelic variation is a project that's been run by a PhD student in my lab called Pradeepa Pushparai. And she's done a really great job with this cohort that we followed. And we started this early in 2020 when we first got samples from hospital staff that had been exposed to SARS CoV-2. And actually, Dennis gave a kind of a nice introduction for me yesterday in talking about antibodies from SARS CoV-2. So you have a good background already, but I will tell you what we saw in our cohort. We waited for a while before we isolated memory B cells and cloned antibodies. And the reason for that was that it was already known that you will get better antibodies if you wait a little bit, because affinity maturation takes time. And affinity maturation actually goes on for quite a while after the acute infection, even after you have cleared the virus, presumably because there's enough antigen around to still drive the germinal center reactions. So we had a cohort of 14 staff, hospital staff that we sampled. We did the usual thing with looking at antibody titers and neutralization. Yes, they all were positive for spike, they neutralized at different levels. And then we did personalized genotyping of these persons. And that's something that we do, as I described, by generating IgM libraries, 2 independent libraries from each donor. And we use IgDiscover to infer the germline alleles. And so we got 14 different genotypes. And then what we also did was that we sampled memory B cells and sorted them, single cell sorted them for antibody cloning, which we do using the single cell RT-PCR from the sorted cells. What Prady did here was that you can see the gate here of spike-specific memory B cells, and she pulled out 177 spike-specific VDJs. This is the gene usage of these VDJs, and compared to the person's IgG repertoire at the same time point, so the total IgG repertoire, compared to these 177 spike-specific VDJs. So this gives you an impression of if there's a bias in gene usage against this antigen. A couple of things stood out. We had a lot of 169 users, as is often seen. And we also had 330-3 that seemed to be quite overrepresented in these 177 spike-specific VDJs. And we thought this is a good opportunity to learn something, because both of these gene regions are known to be highly variable, both due to allelic variation, as I showed you before, but also due to copy number variation. So here we have 2 gene regions that we know vary a lot between people. And if they are both involved, if genes from these regions are both involved in making COVID antibodies, it could influence how our individual response to the acute initial infection. Of course, you make a lot of other antibodies as well. You make antibodies using almost every other type V gene, and the response is extremely polyclonal. So these 177 VDJs, they were almost all unique rearrangements. So we cloned these antibodies, and this is a list of the best neutralizers we got from this set. And we got some really potent antibodies. So it's exactly like Dennis said yesterday. It's actually quite, almost everyone does generate potent neutralizing antibodies against this virus. And you don't always see such potent responses when you look at plasma neutralizing responses, but when you isolate the antibodies and look at them individually, they're there, and they will continue to affinity mature as you're getting vaccinated and boosted, etc. But we honed in on the genes that I mentioned to you that we thought were particularly interesting. So 169 and 330-3. We had one 330-3 antibody that was very broad, including neutralized the sort of variants of concern. And then we had one that was less broad, but very potent, called CAB-147. And that's the one I'm going to dive into a little bit for a moment. And as I mentioned, these gene regions are quite diverse between people. So we decided to take our study, our cohort, we went back to the cohort and looked at what do these regions look like in these individuals. And we had 8 of the 14 individuals that had heterozygous J or D segments. And that allowed us to use those as anchors to do this inferred haplotype analysis that many of you will be familiar with. This is really nice because it really separates out very clearly which V allele is on which chromosome. And you can see this illustrated here as a cartoon. The haplotype plots are a little bit more difficult to see, so I present this in the context of cartoons here. But you can see that everyone, if you look at the 330 region, each individual is different from everyone else. We have, for example, 1, 2, 3 individuals here that are homozygically deleted for 330-3. This is quite common in the population to be either hemizygote for that gene or have sort of a homozygous deletion. And this, of course, means that those individuals cannot make those types of antibodies that some classes of antibodies. But it could be potentially replaced by other 330 family members that make similar antibodies because the alleles and genes in that class are quite similar. But what's striking when you look at this sort of variation is that, you know, it's the unique sort of character of the loci. If you lack 330-3, you often lack the neighboring genes. This has been reported before, but it's very clear that you sort of lack a whole chunk of that haplotype. If you look at the 169 region, we see also a very sort of great variation between the individuals. And we see many, many persons that have more than 2 alleles, which means that they have had a duplicated 169 on one of the chromosomes. This is shown, you cannot read this probably, but amongst the 8 donors that I'm showing you here, you can see that people have, you know, any combination of 01, 02, 06. We have a novel allele star 20 that was just entered into IMDT. That's from a study by Gurjari and others. That is a novel 169 allele that now has an IMDT name, star 20. We found that in 2 of our donors, I believe. And it's possible that that allele is quite frequent in Scandinavians because Gur's population was also Scandinavian that he studied. But in any case, there is a lot of variation in the 169 region as well as in the 330 region. If we then look at the donor where we clone the antibodies from, this donor had 61 alleles in total, and it had 3 169 alleles. It had 01, 02, and 20. And 2 of these were on the same chromosome. So we thought this was a nice donor to sort of look closer at, specifically since we had also cloned antibodies that use these different genes and these different alleles. And this antibody, CAB-147, was one of these antibodies that used the novel 169 allele star 20. And it was a very potent neutralizer. So we decided to look a little bit more closely at this antibody, and we made germline-reverted versions of the antibody. It had, you can see there are a number of SHM mutations, but not a huge amount. I think it's 1, 2, 3, 4, 7 or so that we in the V gene. And if you back reverted those to the star 20 germline allele, you basically can compare how much does SHM do to make, you know, increase the affinity or the neutralization capacity of this antibody. And this is shown here. So the dark curve is the mature antibody, and the back reverted antibody is the light blue. And it loses activity as expected because SHM clearly helps to improve affinities, but it's still a very potent antibody in its germline reverted form. That's high IC50, about 0.1. So it's still a good antibody. And I think this really says that we are making antibodies directly out of the naive germline sort of B cell population, which is likely important in the early control of the virus. And everyone will do this to different extents, probably, because we all have different alleles. But in terms of kind of, you know, being able to mount a good neutralizing antibody response, you can see that for this individual, it was, you know, one of the ways this person did it was by using this allele. But did it matter? Was it important that it was star 20? So that's the next question we asked. So Praddy looked at the most common 169s. There are many, as I said, but we took the most common ones in the population. Again, we looked at various studies and decided to look at 01, 02, 04, 06, 10, and 20. And we made basically antibodies where we reverted the V gene to all of those germline alleles so that we could look at the impact of allele usage in the context of this antibody. And star 20 was the natural allele, and the others are very similar. You can see in the alignment, they don't differ that much. They have a few coding changes. The most closely related allele to the star 20 is 04, and there's only one coding change in the CDR2, and that's an F to L change. And in all of these different 169 alleles, there is about 5 or 6 positions that are different, but you have different combinations of those different mutations, but you don't find variable positions in other places. But you get quite a lot of permutations of these, resulting in all these different alleles. So having replaced the heavy chain with all of these different alleles, we expressed them, and we basically asked how were they neutralizing now? And again, you see the mature natural antibody in dark blue, and the germline-reverted to star 20 in light blue. The one that was diverted to 04, which only has that one single polymorphism, really lost activity. So it really shows how that one residue made a huge impact on this antibody. And the other ones had no activity. So it's very clear that even a single polymorphism can make a difference in terms of how potent your antibody will be. There are examples from the literature, particularly for flu, where this has been shown, and actually it affects the same mutation that's involved in flu antibodies. So it's interesting to see that it matters for at least 2 different viruses, which allele usage you have. This was very clear, and when we looked at the affinities, it sort of reinforced what we saw in the neutralization, in that the 2 left graphs is comparing the mature antibody and the germline-reverted to the natural allele, and you can see that the on rate is, it's a little bit reduced, but it's pretty good even for the germline-reverted antibody, but the off rate is incredibly more improved with SHM. So SHM really does give you a much slower off rate, basically, and increases the affinity quite a lot. If you compare the left top graph with the right top graph, that's basically the mature antibody with or without that mutation that was different between the alleles in the CDR2. So that also makes a huge difference in the context of the mature antibody that has all the other SHM. And if you compare if you look at the affinity of the antibody that was germline-reverted to the closest allele, 04, it's basically almost a non-binder. So it really makes a difference. And if that person had encoded 04, presumably it would never have gotten this particular VDJ arrangement started. So we think this is an example showing that our personalized immunoglobulin genotype really does dictate how we respond to infections and vaccinations. There are multiple, I'm sure, examples of this one could find, but this is one concrete example. I will shift to discussing inheritance patterns. That sounds grandiose, and there's a lot more to do here, but we asked this in the context of a study group that we had, and this was, why did we do this? Because we've always been intrigued by this enormous variation in the IDH locus. And I think we will hear a lot more about that from Oscar today. It's remarkably high, much higher than for the light chains. It's also higher than for the TCR regions. And why is that? The position of these regions are shown here. The IDH locus is right at the telomeric end of chromosome 14. Is it possible that that's a genomically unstable region? Is that why the IGHV is particularly variable between people? So we looked at a family of 5 where we had samples from 2 parents and 3 offspring. These were adult offspring. That picture shows, looks like they're not, but basically we all had consent for them and everything. So we were lucky because both parents were heterozygous with J6. And 2 of the offsprings were as well. And then, the third offspring happened to have a novel IgD310 allele, which meant that we could haplotype that person too, because we could use that D segment as the anchor. So all family members had heterozygous Js or Ds, which allowed us to do the haplotyping. And this is basically the haplotypes that just listing all the V genes that we see when we do our inference and our sort of core count and our various methods that we use to confirm the genotype of the individual. You can see parent 1 and parent 2 and the 2 different chromosomes for each of the parents that they've inherited from their parents. And they're colored in different colors here. And they're listed in the order of the kind of how the genes are located in the locus. You can see that parent 2 had a large deletion on 1 chromosome, almost 300,000 base pairs. This has been seen in other people. It's not such an unusual thing to have large deletions. This parent also on the same chromosome had a novel 169, another one novel 69 allele. That's indicated in pink here. So when we looked at the offspring, we first do kind of the sanity check to see that the offspring doesn't have any alleles that are not coming from either of the parents. And that was the case. And then we looked at the genotype of the child. And in this case, it's clear that the offspring had inherited the yellow chromosome from parent 1 and the blue from parent 2. So that was all kind of all expected or easy to interpret. When we looked at the next offspring, we again have the 2 parents. We did the sanity check, no alleles in offspring 2 that are not coming from either of the parents. But now we see this pattern, and this is a very clear example of that the purple chromosome has been inherited from parent 1. That's the one that we haplotype with J603. But the other chromosome is a mixture of the 2 haplotypes from parent 2. And you can see that this pink 169 allele is present, but the deletion that was present on that blue haplotype is not. And I'm just going to zoom in on this here and see, so you can see this maybe a little bit better. It's hard from the back, I think, but basically we see that this novel 169 allele was passed down to the offspring, but the deletion was not. So basically this kind of was repaired in this offspring. And this is sort of, we've only looked in one family case, so it kind of suggests to us that perhaps this is not uncommon. And what we think is going on, oh, sorry, just to show the other offspring as well, here it was kind of as expected, 1 haplotype inherited from each parent. But what we think is happening is that you have shuffling of haplotype blocks that occur during meiotic recombination events, and that splits up the IDH haplotype. And this could be one of the sort of something that really contributes to the unusually high variation that you see in this locus. If you have these shuffling events fairly frequently, it will definitely increase the number of different sort of haplotypes you generate. So this is something we're looking more closely at. And from this, then I want to move on to TCRs for the rest of the talk. We were interested in TCRs. It's sort of in a way easier to do inference for TCRs because they don't have SHM. So we thought this was important just also for us to show how we kind of generate these data, the tool. We were very interested in having better knowledge of TCR alleles because that's, as for BCR, has been limited before. So again, this study was led by Martin Corcoran, and here he was greatly helped by Mark Cheryshev, again, who's here if you have any questions, and another PhD student, Marco Mandolesi, who also worked very hard on this study. We used these population groups that are shown on this slide, and we generated libraries for both the TCR alpha, beta, gamma, and delta genes. We ran IgDiscover and all our different sort of methods to confirm the genotype in each of these individuals. And what we find here is shown in these trees here. In total, 158 novel TCR V alleles. And they distribute, as I show below, 77 for the alpha chain, 65 for beta, 15 for gamma, and 1 for delta. And as some of you will be aware, Gurjari had a paper where he used IgDiscover for TCR inference for the beta chain, and we do see several of the same alleles as his. I think he had 24 alleles novel in that study, and the majority of those we find as well, not all. And then we find additional alleles. We had a bigger and more diverse population group. And again, most of these are coding, not all, but you can see about 2/3 have coding changes. And yeah, several interesting observations. One of the things that may be worth mentioning is that 24 of these alleles overlapped with alleles that were present in IMDT, but they were truncated in IMDT, so they were not full length. But we generate the full length allele, so we can kind of correct the missing information from IMDT. So, but that was just 24, and the rest were not completely novel. In the, when it comes to the J chain, we see also a lot of novel allele, 24, that distribute like this, 17 for alpha, 4 for beta, 2 for gamma, and 1 for delta. And here, they were almost all coding. And I think this is potentially very important because a lot of these coding changes were right up in the 5' end of the J gene, and that can affect the CDR3 in many cases. So having these in the database is sort of important for understanding, looking at TCR repertoires, clonality, etc. And we had a set of 45 donors, as I said, but we realized that we could extend this to a larger population by identifying SNPs that were unique, that characterized an allele. Once we know the full-length allele, we can look for combinations of SNPs that are only found in a certain allele. And by doing that, we could use the 1,000 genome samples collection, and we could basically then extend and look at whether this sort of frequency of alleles in different populations hold up. So we did that. We could identify from the 1,000 genome sample collection donors that had the potential alleles we were interested in. We went into those samples and PCR amplified them and Sanger sequenced them and confirmed that the alleles were there. And then we calculated the frequencies of the given alleles based on SNP data in 2,504 samples from the 1,000 Genomes Project collection. And this allowed us to sort of confirm our population frequencies. And I'm going to show you how it looks. And this is sort of one of the slides. The way to look at this now is that for each allele, there's 2 graphs. So these 2 is 1, etc. So you're always looking at our cases to the left and the 1,000 genome samples from the bigger sample collection on the right. And then we're plotting this as frequencies. And you can see that there is a really high concordance between, so some alleles are only found in East Asians, others are only found in South Asians, many alleles are only found in Sub-Saharan Africans, etc. So there was a really good concordance, and this kind of gave us confidence that our frequencies were right. We also were able to get information on an additional population group because in the sample collection, we also had Americans, and they are, they have representation from several of these groups, and that's clear from the data. That's looking at the V genes. And if we then look at some of the other here, showing alpha J, gamma V, gamma J, delta V. This is just an example of that. We did do this for all of them, but I'm showing you some examples. And again, the concordance between our data set and the large data set is very high. We were sort of interested in these alleles that seem to have. It's not unexpected that there is a lot of alleles in Sub-Saharan Africa that are not present elsewhere, because what left Africa, when humans left Africa, there was just a sub-selection or subgroup of people who left the continent. But there's also these alleles that are only found in Asians and Europeans. And an example of that is found in TRGV4. TRGV4 has quite a high allelic variation, 8 alleles. And one of these is very clearly present and actually very frequent, both in Europeans and in East and South Asians. So somewhere like around 10% in those population groups. And when we looked at this, more carefully at this particular TRGV4 allele compared to some of the other TRGV4 alleles, you can see that it encodes 4 coding changes. And this actually has 8 nucleotide changes, and 4 of them are coding, and they're in the CDR1, 2, and in this hypervariable region 4. And this is a very divergent V gene. So Martin basically looked at how it looks, how it sort of looks in comparison to non-human primates, and he looked, and you can see here that I can do this. I think if you look at the Homo sapiens TRGV4 alleles cluster here, we have a gorilla, a bonobo, and chimpanzee, and then this novel one that's clustering along with these sort of ancient relatives. So that really suggested to us that this may be, an old allele, this led immediately us to look at the assemblies of archaic humans, and this is information that's extremely valuable, data generated, and particularly by Svante Pääbo, where we have 3 full-length Neanderthal genomes and 1 Denisovan genome. Mark here did this work, and it was clear that every position here was identical to these archaic, what you found in these archaic genomes. It was the same positions and the same nucleotide difference. So to sort of cut a very long story short, we were able then to show that this is in fact has entered the Homo sapiens population through an introgression event, and that this haplotype was derived from our archaic relatives. And I am not showing you all of the sort of work behind that, but we collaborated with Mattias Jakobsson at Uppsala University, who studies who does this kind of work, and he basically worked with Mark to apply all the kind of appropriate statistical methods to show how large is this introgressed haplotype, and to sort of provide evidence for that. And when we look in the 1,000 genome samples, we see this allele in, as I say, about 10% of the cases. And you really do wonder, what does this allele do? Has it been positively selected? Does it have a functional role when it's present to that sort of level? And what's known about this with TRGV4? There is a literature on this. TRGV4 has been shown to interact with butyrophilin-like molecules. These are classes of proteins that have a role in kind of homeostasis and immune surveillance. Adrian Hayday in London has done a lot of beautiful work on TCR gamma delta T cells. And in this particular paper that was published in Cell, they show how TRGV4 interacts with these ligands. And he shows that they're germline encoded amino acid residues that are important for this interaction. So it's not a CDR3 interaction, it's germline encoded. And the proposed interaction sites is very, very close to where we have polymorphism. So the next step for us is, of course, to understand how this allele, how TCRs that use this allele interact with these ligands. And this could have impact or it might be of relevance, for example, in celiac disease, because these intraepithelial lymphocytes are often, there's an accumulation of TCR gamma delta T cells in this compartment. And so this is sort of showing you the population kind of a restriction of some of these alleles. Again, just like for IGH, we see many alleles that are only found in sub-Saharan Africa. We see quite a few that are only found outside of Africa. So the picture is really similar between these groups. Now, I haven't looked at the clock, but I will summarize and say that we strongly feel that individualized genotyping of both the BCR and TCR V DJs really highlight the allelic diversity and is important as we go forward in various settings to look at functional responses. Otherwise you miss sort of the devil is in the detail. If you don't know what allele the person has, you may be missing things. And we have an example in the COVID work that I showed you that this does matter for functional antibodies to generate certain types of neutralizing antibodies. We have discussed the fact that the IGH locus is particularly diverse, and this may be at least partially explained by these shuffling events that we think we have evidence that they occur during meiotic recombination to create new haplotypes. And finally, we have archaic introgressed events that contribute to our diversity. And this is really kind of, it's a way for what this happens because, if they have an advantage, they will be maintained. And the frequency of this particular allele that I spoke about suggests that it's doing something that we're very interested in. So I'd like to acknowledge my group, and everyone's working very hard on this. Several of the projects I mentioned are led by Martin Corcoran, and also excellent contributions on many of these projects from Sanjana Narang, Mark Chernyshev, Marco Mandalesi, Matthews Kaduk, Pradeepa Pushparai, Andrea Nicoletti, Sungyong Kim. And for the COVID work, we collaborated with Dan Stewart and Ben Morell, and as well as people in Jerry McInerney's group. Some of the samples from sub-Saharan Africa were obtained from the Fan at Sundling labs. And for the archaic alleles, we worked closely with Matthias Jakobsson's lab at Uppsala University. Thank you.
Microphone. Get out. That's a really fascinating talk. Thank you so much. Again, probably ignorant questions, but do you think this implying that there's like co-evolution with pathogens in these territories?
I think that would, of course, make a lot of sense. That over long periods of time, there's been pressure or exposures that have shaped our these loci. So I think I definitely expect that to be the case. It's always hard to prove that because we don't have access to those kinds of samples. But as we understand more and more about how certain V genes are used in the response to different pathogens, I think we will get clues.
Yeah, I'm wondering, like doing functional tests of some of these specific alleles against known pathogens in regions and seeing whether those germline alleles naturally generate good neutralizing antibodies against.
Yes, and I think it's clear that I think we really can straight from from the output of the naive B cells often make fairly good antibodies, and the rest is kind of refining those. Right. But I mean, we have evolved these loci over millions of years, so it's clear that there's been time to evolve. And I mean, the integration event is something that must have happened about 50,000 years ago when we coexisted with Neanderthals and Denisovans. That's more kind of you can kind of home in on a period there because it's not that long that we lived coexisted.
And I don't want to hog the show, but touching on your last point, you think there could be some relevance here to autoimmunity?
So I think there could be. I mean, that is, whenever we think about the adaptive immune receptor variation, there's always two sides of the coin, for sure. There is our ability to respond to infections and our ability to avoid having autoreactive responses. But most of autoimmune diseases are, kind of not equally represented in different parts of the world. So some are very common in Europe, for example, and others are not. And I do think that there are going to be these predispositions. I think a lot of autoimmune diseases are complex. They are multifactorial. But you could have an allele that does push you a little bit one way, and that together with an MHC haplotype and something else, a trigger, could be enough.
Yeah, and I'm thinking of people who've been explanted, like if you're, say, African American, whether you're exposed to things that you might not be in your typical environment, so on. That
definitely possible.
Yeah. Fantastic talk. For the second story, where you look at the variation of the alleles and their binding activity, did you look also at the structure, or are you making crystal structures to look at how the structure influences this?
Yes, we have, for this antibody, we actually have EM data now. I mean, cryo-EM high resolution. So we can see, this, this position, phenylalanine is really part of the contact. Yes, so that's clearly visible from the complex, the structure.
And second, do the alleles influence the antibody structure itself? So is the structure different?
We don't have structural information for all those variants yet, okay. But that's an interesting question. Right. Yeah.
Thanks, Gunilla. Thank you. So very nice, and thanks for working on those TCR variants. Do you know if there is any association between the variants and the usage of the different genes? If there is an impact at the end of on the global repertoire?
Right, I think that is the case for some of the genes, and we know that is the case for some genes very clearly. You know, we've looked at it for some of them, and I think Oscar is going to refer to this in his talk as well. Okay. So, you know, because there's also polymorphisms outside of the coding region that could affect this. There could be that some of these polymorphisms that are in the coding region, you know, impact, say, negative selection, that something is not tolerated. If you have a big, you know, bulky amino acid in some, so you may get less of those B cells surviving and becoming naive, mature cells. So those things are, you know, I think really interesting, and there is, as we know, you know, 100-fold difference in gene in frequency use between the most used B and the least used. So there must be a reason for that.
Thanks.
Very nice story. Regarding the information content of IGH as a whole, I think we've all come to accept the fact that a complete IGH genotype might be identifying, but given the recombination story, would that already apply to an individual haplotype? Or have you seen in your larger stories haplotypes that are shared, so individual loci?
So, I mean, I don't know if I completely heard your first part, but the examples with this kind of, you know, acquiring a part of the locus from two different chromosomes of a given parent, that we have, you know, this example, I think, you know, as we look at more families, we will see. But you were asking something else, I think.
No, so this is pretty much the question that I'm asking. Would you expect to see the same haplotype in people that are not related to each other?
Yeah, I think, I mean, like, there's probably some limit in how many different haplotypes there are, even though it is extremely diverse. Yeah, I think we just have to look when we start having data from, you know, more individuals.
Okay, but at this point, you cannot yet put a number on this.
No, I don't think I can. I think it's, you know, we do see some things that look kind of similar, but yeah, I mean, even this big deletion that happens in some frequency of people is not identical in the, you know, it's kind of roughly in the same region, but it's not the genetically identical.
Yeah, okay.
So there's probably some regions that are just prone to this.
Mm-hmm.
Yeah, I think, you know, once we have more genomes and genotypes, we will learn about this.
Okay, thanks.
So with the discovery of all these novel alleles, does that mean that we've been overestimating somatic hypermutation because we've been comparing against the wrong germline allele?
I think so. I mean, I think that is, and you know, we've done a lot of work on this in the macaque model where the problem was much bigger because the databases were missing genes, not just alleles. And then you have, you know, this big discrepancy. If you assign to the next closest, you have, you know, you really get wrong and very much inflated SHM. In humans, yes, it's going to be, you know, ideally you want to assign it to the right thing, right? So I think the improved databases or the personalized genotyping is the only way to do that. But you know, it's not going to be, yeah, it's going to be a few, you know, nucleotide or, you know, a smaller percentage of kind of errors, I would say. But definitely one should aim to have it correct.
I have a question from a remote attendee. So Corey Watson is asking, well, is saying first, Nila, amazing talk. How do you or we get groups studying BCR and TCR responses to more formally integrate germline information into their studies?
Yeah, I mean, I think, you know, we're trying to get it on the radar, first of all, that, you know, allelic variation is important. You know, a lot of studies, classic immunology studies and antibody cloning papers, they just assign to gene, they don't assign to allele. So it's not so precise. But I think if people start assigning to alleles, that's already, you know, we will start seeing more examples of how this could matter. And in the meantime, yes, I think if, you know, for those who have the possibility to look at a specific antigen, specific response and do, you know, do this kind of combined functional and genotypic analysis, it's definitely doable. We have another study where we're looking at autoimmunity in this particular case, rheumatoid arthritis. You know, we have good sample collections at Karolinska. This other, you know, we are very interested in working with the clinicians that have those kind of sample collections, so we can look at sort of larger cohorts and start to understand how this really potentially impacts, you know, the development of a disease, for example.
Okay. I was wondering if you have looked at genetic variation within BTLN to see if there's like some co-evolution of variants between that protein and the TCR allele that you observed? We haven't done, you know, we really haven't had time to do anything in terms of following up that interaction, but it's so obvious we have to, you know, just find time to do that. And I think it is really interesting. The ligands are also variable in the population. Yeah. So there's more, you know, both aspects could be variable. So if you have a certain combination of ligand with a certain TCR V4 allele, you know, that sort of thing.